The question of how life first sparked into existence on a young, barren Earth is one of science's most profound and enduring mysteries. For centuries, thinkers and scientists have pondered this enigma, trying to piece together the incredible journey from simple non-living chemicals to the first living cells. Understanding this origin story is not just about satisfying our curiosity, it's fundamental to understanding what life is, how it evolves, and whether it might exist elsewhere in the vast universe. Scientists approach this by studying ancient rocks, experimenting with chemical reactions, and looking deep into the genetic code of modern organisms to find echoes of the very first life forms. It's a detective story on a planetary scale, spanning billions of years. For a long time, a prevailing idea in the scientific community was that the essential building blocks of life, particularly amino acids, appeared in a more or less defined sequence. Amino acids are vital molecules that link together to form proteins, which are the workhorses of our cells, carrying out countless critical tasks. The thinking was that simpler amino acids likely formed first from the primordial soup of chemicals on early Earth. Then, over time, more complex amino acids would have been synthesized and incorporated into the developing machinery of life. This stepwise addition seemed logical, reflecting a gradual increase in biological complexity, a slow climb up the ladder of life from simple beginnings. To understand this further, it's helpful to know what amino acids truly are. Imagine them as different types of Lego bricks. Each amino acid has a unique shape and properties. When these bricks snap together in long chains, they build an incredible variety of structures called proteins. These proteins might become enzymes that speed up chemical reactions, or they might form the structural parts of a cell, or even act as messengers. Life as we know it depends entirely on the precise sequences of these amino acids creating functional proteins. There are 20 standard amino acids used by nearly all life on Earth, and scientists believed their emergence followed a specific, predictable order. Central to the study of life's origins is the concept of the last universal common ancestor, or Luca. Luca is not the very first life form, but rather the most recent organism from which all living things on Earth today have descended. Think of it as the great-great-great-grandparent of every plant, animal, fungus, and bacterium. Luca likely lived around 4 billion years ago and already possessed a relatively sophisticated cellular structure and genetic system. By studying the features common to all its descendants, scientists can infer what Luca might have been like and, critically, what kinds of proteins and amino acids it already used, providing a benchmark for what came before. Recently, a team of researchers from the University of Arizona decided to take a fresh look at this long-held idea about the sequential appearance of amino acids. Their study, published in June 2025 in the Proceedings of the National Academy of Science, aimed to investigate the types and frequencies of amino acids present in proteins that existed even before Luca, as well as those that came after. This ambitious endeavor sought to reconstruct the very ancient history of protein evolution, hoping to confirm or challenge the established timeline of when each amino acid joined the repertoire of life. It's like being an archaeologist of molecules, sifting through ancient data to uncover our deepest biological roots. The scientists, led by senior author Joanna Mazel and first author Sausan Vaibi, employed sophisticated computational tools to conduct their investigation. They utilized vast databases of protein information, specifically data from the National Center for Biotechnology Information. Using specialized software, they constructed an evolutionary tree of protein domains, stable, functional parts of proteins. By analyzing these domains, they could infer which amino acids were common in the very earliest protein structures, dating back to the era before Luca, and compare them to the amino acid usage in proteins that evolved after Luca. This method allowed them to quantify and compare amino acid presence across these pivotal evolutionary periods. The results of their painstaking analysis yielded some significant surprises. One of the most striking findings concerned an amino acid called tryptophan. Tryptophan has traditionally been considered one of the last, if not the last, of the 20 standard amino acids to be incorporated into the genetic code and used in proteins. It's a relatively complex molecule, and the assumption was that its synthesis and integration into life's machinery happened late in the game. However, the Arizona team found evidence that tryptophan was actually present in significant amounts in the pre-Luca data, at about 1.2%, compared to 0.9% after Luca. This early appearance was unexpected. This discovery about tryptophan, and potentially other amino acids, directly challenges the established timeline for how life's building blocks came together. If tryptophan was around, and being used much earlier than previously thought, it suggests that the story of amino acid emergence is more complex, and perhaps less linear than the simple to complex model proposed. The 25% higher prevalence of tryptophan in pre-Luca protein domains indicates it was a more integral part of early biochemistry than scientists had appreciated. This finding forces a re-evaluation of the chemical conditions and evolutionary pressures that shaped the very first proteins, and, by extension, the earliest forms of life on our planet. The implications of these findings are quite profound for our understanding of how life may have started. The previous model, which suggested a clear order of amino acid emergence based largely on their perceived simplicity or ease of abiotic synthesis, might have been an oversimplification. The Arizona study suggests that perhaps the frequency of amino acids in early life forms was not the sole determinant of their emergence order. It's possible that early proto-life, the very rudimentary precursors to true cells, was more sophisticated or utilized a different set of resources than we previously imagined. This new perspective opens the door to considering a wider range of possibilities for early biochemistry. Furthermore, the research hints that the primordial soup, the classic image of a planet-wide ocean teeming with life's precursor molecules, might not have been a uniform environment. Instead, different amino acids could have originated from various distinct environments on the early Earth. For example, some might have formed near volcanic vents, others in shallow pools exposed to ultraviolet radiation, and perhaps some, like the aromatic amino acids, which include tryptophan, could have formed at the interface between water and rock, such as in alkaline hydrothermal vents deep in the ocean. This mosaic of environments could have contributed different building blocks at different times or in different concentrations. This complexity extends to how the genetic code itself might have developed. The genetic code is the set of rules by which information encoded in genetic material, like DNA or RNA, is translated into proteins. The Arizona study supports the idea that the construction of this code might have been a stepwise process, but also that there could have been competition among different ancient codes simultaneously. Early life might have experimented with various coding systems, and perhaps even utilized non-canonical amino acids, those not among the standard 20 we see today. These ancient codes could have emerged and evolved in specific niches, like those around hydrothermal vents, before a more universal code eventually won out. The dynamic nature of origin of life research is further highlighted by other recent studies. For instance, research published in late 2024 showed that RNA, a key molecule in early life theories, doesn't inherently select for only left-handed amino acids, which are the exclusive type used by life today. This suggests early life might have used a mix, and evolutionary pressures later refined this choice. Another study from mid-2024 proposed that lightning on early Earth could have been a significant source of reactive compounds needed for prebiotic chemistry. These diverse findings, including the Arizona study, paint a picture of multiple plausible pathways and a much richer, more varied chemical landscape for life's beginnings than previously conceived. The insights gained from rethinking the emergence of amino acids on Earth have significant implications for the search for life beyond our planet. If complex amino acids like tryptophan can form abiotically and be incorporated into early biological systems more readily than we thought, it widens the range of conditions under which life might arise. For example, the University of Arizona researchers suggest that the abiotic synthesis of aromatic amino acids might be possible in the water-rock interface of the subsurface ocean on Saturn's moon Enceladus. This icy moon, with its hidden ocean and potential hydrothermal activity, is considered a prime candidate for harboring extraterrestrial life. This expanded view also informs our search on other celestial bodies. Scientists are already observing lightning and plasma-induced chemistry on planets like Jupiter and Saturn, and potentially on exoplanets orbiting distant stars. If such processes contribute to creating life's building blocks, then the potential for habitable environments and the emergence of life elsewhere could be greater. Furthermore, the analysis of amino acids in meteorites, such as the famous Murchison meteorite, and in samples returned from asteroids like Bennu by NASA's OSIRIS-REx mission, provides direct clues about the types of organic molecules available in the early solar system. Future samples from Mars will also be scrutinized for these vital compounds. Ultimately, this new research from the University of Arizona, by challenging old assumptions, brings us a step closer to understanding our own origins. The story of how life began on Earth is our story. Each discovery, each refinement of our theories, fills in another detail in the grand narrative of our planet and its inhabitants. Realizing that the path to life might have been different, perhaps more varied and opportunistic than we imagined, doesn't diminish the wonder of it. It enriches it. It underscores the incredible journey from simple chemistry to the complex biology that surrounds us and that constitutes us. Science thrives on questioning, re-evaluating, and building upon previous knowledge. The origin of life remains one of the biggest unsolved puzzles, and as research like the Arizona study shows, our ideas about it are constantly evolving. While a complete step-by-step scenario from non-life to Luca is still being pieced together, each new finding provides crucial pieces. It shows that even deeply entrenched theories can be revisited and revised in the light of new evidence. This ongoing process of discovery not only refines our understanding of life's beginnings, but also fuels our quest to find our place in the cosmos.